was told that I should give, since I had initiated this area in India as before in 1970s, particularly in 74, we first presented some work on it. So I have going to talk to you on computer drug design and discovery, and Odyssey means a kind of a journey, right from its initiation till now. As you all know that you are seeing two pictures on my introductory slide. One is of CDRI, where I have spent more than 50 years. Only on 16th of July, I have relinquished my emeritus scientist position there. And I am now chairman of the institute. This is called Global Institute of Pharmaceutical Education and Research. It is in the birthplace of mine, which we have founded at about 9 years back. So all the work which I have done is I'm going to take, in fact, to you right from how a medicinal chemist, when I joined CDRI somewhere in 1969, then at that time, we were just research fellows working on synthetic chemistry, of course, with the medicinal point of view with Dr. Nityanand, who was my mentor. And at that time, what happened was that people used to go to the project meetings, and it was always there which activity has to be. So everybody was worried about activity, and people were getting those compound screen, which also people do now. So I was always thinking activity prediction part of thing. And later on, during my even PhD fellowship time, I came to know something is going on prediction. And I read some papers, particularly Corwin Hunch paper of Journal of American Chemical Society of 1964. And I thought there is something, how the equations come. And I contacted some person in statistics department, Lucknow University, and totally of my own, because at that time, no one in CDRI was knowing about these things. So I wanted to discuss, but people said, no, we don't know, but something is coming like this. And I started doing without calculator even. At that time, I didn't have the calculator also in 72, 73. So I started doing and put the equations and learned it. But thereafter, I think it was 1976, a drug design symposium in which Corwin Hunch also came, and I was asked, people came to know it's my kind of a hobby, and Dr. Nityan asked me that, can you do something in this conference, because no Indian is there who is going to speak on this. So I made 2 research papers and I presented there. And that was the real rise, where I met so many international scientists like Corwin Hunch, Goodford, and Topless, and many top guys on this field. And they came, Professor J.K. Seidel, with whom I worked as Humboldt Fellow also. They all came here. And I got exposed to computer-aided drug design. So today I will give you a brief introduction first about what is generally this drug design and discovery research is, and then I will tell you the whole problem, how I initiated this, a normal one, and then went ahead. As you know that why it is so difficult to make drugs, because if 10,000 chemists prepare one compound per second, then it will be 10 raised to about 27 years will take to finish the job. You can imagine how difficult. So you cannot explore the whole chemical field through just by taking the physical compound synthesis. And the history of drug discovery goes in things repeats, it goes in circular way mostly. But drug research history doesn't go in circular way, rather it goes in a kind of a helical form. And each time it goes deeper and deeper. If you see here the traditional knowledge base or random screening, which was right from the beginning, people having the plant products or people having just general screening, it went further to give a rational design, random screening. Then came structure-based drug design and indirect design, combinatorial chemistry, HTS, informatics, that is chemoinformatics, pharmacogenomics, all this. And the whole drug discovery challenge, I think you all know it, that it takes very long time for a drug to discover. It takes me about 14 years, right from the beginning. And the drug discovery phase takes about 4 years, and then phase 1, phase 2, phase 3 trials. And this is also takes a lot of time. Recently, I've just on the 19th of this thing, 19th of July, I found that now the cost is estimated to be 2.6 million, which used to be, of course, less before this. So I'm going to focus how a medicinal chemist, because medicinal chemistry is the main thing, how one proceeds for it. And what used to happen in a pre-genomics part, the time period for drug discovery phase about 6 to 7 years, which has now been reduced in a post-genomic to 4 to 5 years. A lead has to come. First part is lead generation, which used to come from plant origin, random screening, or biochemical origin, combinatorial chemistry at that time. Then followed by lead optimization step, and which takes study of analogs, SARs, comparison of standard drug activity. But what used to happen before, people were going in this direction, then they used to think of molecular targets. What can be a target? That used to come in last phase in it, in one way, in the drug discovery phase. People used to think, but nowadays when this molecular biology or all this came up, this took a shift that now you know the molecular targets first. We identify the molecular targets through genomics and proteomics, etc. Then we validate this, called chemogenomics. Then, of course, from here we take a lead. On that lead, again, of course, can be from any of these sources, and which is now, I would say, more focus I will give on virtual screening, which is now through computer aided. Combinatorial chemistry on STS has gone out because this was very, very expensive. I will not discuss that much, but it was at that time in 1990s, late 1990s, early 2000, this was very much on. Then lead optimization, where comes QSAR and CAD and so forth. The other phase, which is a drug development phase, there toxicity, phase 1, phase 2, phase 3 clinical trials that come. That time is not very much reduced, but it is almost there, and the investment has gone up. So at one time it used to be far less. If you see on the left-hand side of mine and the right side of the slide, right and left, just you find a difference. Now I say medicinal chemistry is the heart of drug discovery research. Why? The reason is, I have compared conceptually like a heart, because in heart you purify the blood and then it goes to the different section of the body, the parts of the body, and it gets purified. So I take it like this. First you make a compound, chemist makes, tests it, and then some problems comes in vitro. Then again, chemist thinks again purifies, send it when it is active in functional cultural assays. Then it again the same thing. It is not effective in vivo. Then again it does something, modulations in a structure so as to meet the pharmacokinetics or pharmacokinetics part, and so on and so forth. Finally he gets, purifies, then it gets a candidate drug for in vivo toxicological evaluation. Again at that level also, finally again it comes to medicinal chemist, and I don't go in detail, but rather at the time when the drug is going to be released, again it comes to the chemist that you should have a easy process for a synthesis. I would also like to mention here why in India, I'm just making just a comment, we have done very good in part of the process development. You know, what's the reason? The reason has been that India has right from the beginning had given a good emphasis on chemistry, particularly organic chemistry and synthesis part. But unfortunately in this country, even up to this time, the importance to medicinal chemistry has not been given. There's a very fine difference, a border between the chemist and the medicinal chemist. Sometimes the chemists also turn to be medicinal chemists, but they're full of life. They are making the same compound which have shown activity. They go on modifying and mode on modulations, other things they can go on. Certainly the compound will show activity. But the real medicinal chemist is the one who has got two things. Chemist knows how to synthesize, but the medicinal chemist has to know how to synthesize as well as what to synthesize. What to synthesize is a big multi-million-dollar question. There comes lead generation. You can optimize also, but the lead generation is not the part. How a medicinal chemist does it. So he's like an architect who knows also civil engineering, while the chemist is like a civil engineer. Now I will tell you how a normal medicinal chemist, even if he had not idea like our very important ones like Dr. Nityanand or a medicinal chemist who had been here, or Nagarajan, or I would say only one or two are there in the whole of country, which I say real medicinal chemists. So how do people used to think? First of all, they see pattern recognition. What is pattern recognition? Pattern recognition, you see the organic molecules, you see the structures, and you know picture goes inside brain, and you see, oh, this compound was showing activity. So you read the paper, which I also did. You find out some substructures and you join them together in different manner, either substructural approach or medicinal chemical hybridization, or you design a prototype by grafting of these pharmacophores of substructures, assimilate them, then if there is a conformational restrictions, you can rigid them, or you can introduce conformational flexibility, and you can work on the compounds in that area. So the substructural approach, which is qualitatively has done by many, even most of the people who don't know computer-aided drug design, they do it intuitively, and depends on their brain how fast they do it. But the part is there's a substructure activity frequency, I can say in that way, formulated that the substructure, if it is present in 100 molecules and 80 are active, I would say 0.8 is activity frequency, means how much active upon total compounds. And if you put that substructure in a way so that your mean substructure activity frequency goes up, then I think you can find out, design the molecules. Of course, then you can use also the substructure indicator variables in the QSIR studies to define the substructure. Then comes a medicinal chemical hybridization. You see here propylidazine, which is a vasodilator. The propranolol is a beta-adrenergic antagonist. So you want to have both the properties. So you take one part which is responsible for vasodilation, the other part from there on this, you mix them together and pyrazidol synthesize. This compound has shown both activities, but sometimes it may fail also, but it depends what you have identified. I will just give in, because since other part I will deal in detail in my example. About just this example, I'm telling you that I did it, practice it. It was like this, a xanthaquin molecule was there in the CDRI, which of course I had been one of the person involved in this initially. And the other compounds we had. So I found out which are the substructure which is most important for this activity. So we mixed them together and we generated a new novel molecule, 86204, it was 1986. And you will find that this compound was as good as xanthaquin, even somewhat better also. So this is a kind of a new thing where you make a medicinal chemical hybridization. And recently I'm very happy to say that this xanthaquin, which went up to phase 1 trial only as antihypertensive, but has been recently, we have approved it as IND committee member. I was also there in October this time for hypovolemic shock patient. And very recently in 27th May 2020 in Pharma Letters, it has been found that xanthaquin can provide hemodynamic stability for COVID-19 patients. You can have the reference. So I mean to say this is a kind of repositioning of this drug. And you see here the activity, the comparison of xanthaquin with 86204, both in vivo as well as in vitro in binding site. And it is like this. So this is the way how a medicinal chemist designs and gets the results. Now let us come to the computer-aided drug design, which is topic. There can be two kind of drug design. One is, I will say, direct design, where you know the target structure, for which many people are now working hard, and everybody like. But in 1990s, before 1990s or 2000, even today, you are not having the structures of all the targets. There are several targets. So the other approach, which has been very successful and is still, but people are now using it less and less, which I want to emphasize how good this approach is. And I will focus my work also on more on indirect design. That is a ligand-based drug design. Here you don't know the structure of the target, but you know the chemical probes. You know their activity is increasing or decreasing. And from this you analyze. It is just like a person who's a blind and a tailor and who is stitching a cloth where he just feels by his hand that whether it is long or short and feels the body, but he doesn't see the whole thing as usual when one is not blind. And this is this kind of. So what does one do in this indirect design? It is a kind of a molecular structure. You correlate with physicochemical properties or biological activity through its structural properties, structural descriptors, which can be 1D, 2D, 3D. Out of them, you do a selection, and then you make a theory that how it is related to this, called structure-function relationships or structure activity. Now, this goes back as early as 1964. As I told you, this was a hunch. There were two groups at that time, hunch and Free Wilson. Hunch subjected that a drug receptor or an enzyme, drug or a substrate with enzyme or a receptor, X, and there is a drug receptor complex or enzyme substrate complex formed, which gives to a different kind of internal processes a biological response, which you study. And the free energy change of this is directly proportional to the drug concentration, which in turn is related to the electronic hydrophobic, that's the electronic hydrophobic steric effects, and some structural parameters like here I put delta G as change in this free energies of these. Of course, there can be two things. It can go linearly also, which I have shown on the side that It can be linear. Sometimes if it is a partition coefficient, log P, it's going to be parabolic because the compound is not reaching to the active site. As a very optimum partitioning is needed. In our work, when I was doing this in 70s, I also got a chance to write a chapter on progress in drug research on QSARs, and there I combined both these approaches and I put the substructure approach like Free Wilson, where the contribution of the substructures is taken together with the parent one. So I put I1, I2, these are the indicator variables by which you can combine both physicochemical approach and this approach. This is called a combined approach. Now it's used quite extensively. So I would not go in very much detail, but I would say now a virtual screening paradigm can be a multi-level, which is also, you must have been aware with other lectures. You first of all, you just make a library or in-house library or database or focus library. I'll give you some examples. Then you know these properties for admit parameters, because if compound has to go to in vivo, then I think you must have these properties. And you have got similar searching methods. You have got Pharmacophore model. The more you come down, it is more accurate. You can come to QSAR models. And then finally, when you identify, come to a very short layer, say of 100 or 200 compounds, you can do docking studies if you know the target of this and you can validate. And those compounds which are prioritized, you can then synthesize and you can cut short this drug discovery phase in a tremendous manner. And I will give some example too. Now I'm now entering with the work which we did when I joined CDRI, and I think we were given this. So what we did a kind of a substructure analysis. You see in haloperidol, chlorpromazine, oxypartine, CDRI 69183 was compounds. And we found that these are the things. In haloperidol, you have got this substructure, palafluorobenzoyl propyl. In chlorpromazine, you have a benzene. In oxypartine, you have got indole part. And CDRI 18183, also this parafluorobenzoyl, this had got antipsychotic. So we fused them together. And you know, this compound, it looks very simple. This compound is a drug which was released to market from CDRI as early as 1986 by Prime Minister Rajiv Gandhi. And we got a lot of patents. Its compound was approved as a drug. When we did the QSARs on this series of compounds, we found one very important parameter is indicator variable. An indicator is variable is for this substructure where a parafluorophenyl ring or a aryl part is separated by nitrogen, which is called neuroleptic nitrogen, parasite nitrogen, by 4 carbon atoms. And this was the substructure which I show you as identifying. Now, having this theory of substructure analysis, we found that is meaning substructure activity frequency of a substructure where it is many times, it can be increased. So with this 3 times repetition of this substructure, we designed a molecule of CDRI 86373, and really this compound was better than sanpentindole. Of course, it was not went further because at that time we already had the sanpentindole and its enantiomers. But this compound was as good as this, rather it has a better property as compared to even rasparidone, which is a drug. And here the slide shows in vitro as well as in vivo binding of these compounds against D1, dopamine D1, dopamine D2 receptor and 5S2 receptor, where you find that this comes. So this is what the designing is. And here we did also the pharmacophore modeling. It was not done at that time. We were not knowing the dopamine that way, dopamine receptor, but it was. But now we have done also dopamine receptor part. But this is one part. The most important part, which actually brought me to learn QSAR and to also, I want to put it here, how one enters into it. Since in this series of compounds, we get a lot of variation in R, the 2 important variation was of course potent neuroleptic drug. The other one was antihistamine activity. So we went to this substructure, NSCO1, which you see second, where we went further to improve, to dissociate totally, whether neuroleptic to anti. This is also done. This is called side of SOSA approach, where you side observations theory, where you can improve take the lead from the molecule of its side effects, and you can then develop a molecule onto that particular activity. So antihistamine activity. So a very simple QSAR we did, which I did partly with the calculator at the time. You see, these are the molecules which were there, and you see here we have got these number of molecules, and of course their histamine, antihistamine activity in vitro. And here are the different parameters like hydrophobicity, RM value, pi ortho, pi meta, pi mera, and MR ortho indicator value free variables, because diversified this hunch approach goes into one class of congeneric series. So I was also thinking how we can further enhance its scope. And then I used these indicator variables. And finally, these equations were generated for these molecules. And you see here, the hydrophobicity is very prime importance. Apart from this thing, we have got two indicator variables. That indicator variables are for particular type of fragment. In that R substitutions, whole side chain substitution. And there was a one parameter I ortho, which you see in equation 3, that was giving when we are having a ortho substituted compounds in that fragment, they were showing some little higher activity. So we put some indicator variable in order to get a good correlation coefficient. And then I thought, okay, now we know only on these compounds 17, which were having only variation in the aromatic part, we found that ortho position hydrophobicity and para position along with I ortho, which can later substituted by steric parameter MR ortho, was important. But here is, I was not satisfied within myself because it was still a congeneric series, paper could be published, anything, but where are you going from here? So I thought if it is showing antihistamine, anti-allergy, what are the drugs which are known in the market? At that time, diphenhydramine was the one which was very much selling. Still, it is sells in Avil. You see diphenhydramines. One more thing here I want to put here, that in this whole, we had 6, means there were 6 compounds which were not fitting and we had to remove out of the QSARs. And those 6 outliers are here. Here you see their actual activity and you see the predicted activity. There's a large difference, particularly the red ones. There's a significant difference, particularly in the compound 6. The other 6 compound was the one which was considered by pharmacology department to be developed an antihistamine. Its name was, at the time, ethamindole was given because it had a acetamide group, CONS2, the para position of this. And other compound was chlorothiazole, which was number 1. So I mean to say, and so these were outliers, but they were not coming in my QSAR. But I did not leave them, of course, in the equation they are not there, but they were in behind my mind why they are showing activity. Either their mechanism, something is different, some site may be different, but at that time nobody knows even what is the topography of H1 receptor, even what are the amino acid sequencing of histamine receptor is. So this diphenhydramine was the one, and when I compared my results in terms of steric effect of one of the phenyl rings with the diphenhydramine phenyl rings, there was similarity. You must have seen a negative coefficient of ES parameter. So there was this similarity which led me to think that there must be some similarity, and I put a at that time we used to have no computers, nothing, only drying models. So I draw the drying model of our compound and I could show only the complementarity if I fold the conformation of my compound and put on the diphenhydramine, which you see here on this thing side. But actually that was not a how can you fold it? No molecular orbital calculation, no find out something what conformation is. Then one practical thing for a synthetic medicinal chemist would be that I can make a rigid analog of diphenhydramine. So we made rigid analogs. You see here on compound number 4 and 5, rigid analogs. And we found that one rigid analog, which is here, which is the 5-member ring, corresponds to structure 4, that was equiactive to diphenhydramine, which is here. And there was a planar compound also, which was also not that much active. So most active was diphenhydramine, 0.0001 IC50 microgram per ml was IC50 with this. So we made at that time what we thought was that there can be just 4 sites. 2 maybe A and D can be hydrophobic site. There is C can be anionic site, and there can be some, one is called ionizable group where nitrogen is there on B site. So this is what we speculated. And here I want to draw your attention that simple from 2D QSAR, in medicinal chemistry we think, you have to think actually, just for publication, just for this thing, you have to go very deep into it if you want to go, apart from getting the compound, there can be this thing, we postulated that. And taking the assumption, this question came to my mind, that if there is, this structure is there, then what is the necessity of taking indole? We can take a benzene, we have to only have this A, B, C, D sites, then why to have? And I took this assumption that the ligand receptor interaction may be viewed as the association of its component substructures with the complementary subsites upon the receptor and that the prototypes producing similar response may not necessarily have all the requisite substructure elements. Still those common will show. So common subset with different prototype which associate for same subsite would cause similar change in. What does it mean? It means that just like we have two persons, everybody is having hands, but the whole structure may be different. But if your one hand substructure is going to one part in a corner where heat is coming, so both will feel that yes, heat is coming on this side, and initially heat was less, then it was more. But overall heat experience will be different. But this part will be told. So what we did with synthesis, we designed molecules in which we had all these sites, and we took only substructure which was most active and just made variation at ortho position because we know that that was the best compound. And we resembled the QSAR of these compounds. As you see that all those substructures which go to the same site will experience the same biomolecular interaction in terms of the same rate of variation in activity. And you see here the coefficient associated with pi is almost same, 0.304, 0.408, 0.417. These are the different types of compounds. And that shows that, okay, this phenyliding is going at the same site in the biophase. Even for this kind of answer, even if you have a protein crystallized structure, you cannot say what is in biophase. But if you do a genuine kind of even 2D QSAR, it can tell you that, yes, this part is experiencing the same biomolecular interactions. And if you do this variation, this is what this QSAR can lead to. Later on, again, the problem was still how to put them now into something. So I attended one meeting in Germany in 1984, and I met Mr. Marshall, who was at that time working on COMSIA. I met many people there. I met Common Hunch again, and other people like Dovaiko. He was preparing Hazel approach, and I then tried to understand what is Hazel, and this gives you a good concept of pharmacophore modeling to all youngsters. That how does it happen, and how can you put the different diverse kind of structure into the space and can predict the activity. So this was the Hazel work which we did, and it is like this: you take a molecule, you make a kind of a grid around it. You can make 2 angstrom grid or 4, and then have a molecule so that you superimpose each one on these like these type of atoms. You put our best superimpositions, and you develop the Hazel. So I started developing the Hazel from diphenhydramine, which is A, and then putting my molecules. Final Hazel was this. Now this Hazel was the one in which you put any molecule in this space can predict the activity of that molecule. So this was the first one I wanted to identify what is the which is the pharmacophore or important substructure for antihistamine activity. And we could identify that, okay, this is out of the Hazel, this is the best, this is the part. If you put this fraction, I can tell you, take any structure, you put it, it will show antihistaminic activity. So we identified it. Later on, there was a lot of softwares started coming in 90s. First of all, Apex3 we also purchased, which is now in drug discovery, later changed to Catalyst. So Apex3D came, and this is what it shows that this is the site. And you know, there is one very important part was that which is the anionic site. When I put site C, this site C can be given by this nitrogen as well as by the amide part, because it is beyond. If you see from top, it is going down. So I mean, this can be both. So we're not sure. But with this experiment, we came that perhaps it is then NSCO. But then when Apex3D came, I'll not go into detail of this because the software is now also obsolete also. But it did what it did. It took different molecules, 250 conformations, and of that it will superimpose and it will calculate these different properties of this and will develop the QSAR equations. These are the molecules which we put into this. And then it will give you 3 things. One is biophore sites, BS1, BS2, BS3. And there's a secondary site. What are the secondary sites which are in dotted line? That is the secondary site which may not be present and may be present, but that is not the whole real pharmacophore. And that will modulate the activity. And I sometimes say at old times, we used to have a radio station. When we are reaching a biophore, means that sound is coming is Delhi station, but you can do fine tuning by changing the subsites. So I think this is what is there. And this results for these compounds was a good predictive model. And now you can have a diverse compounds, because in this model we didn't have only those count, but you have a diverse molecules, 6-membered, 5-membered ring compounds, and our tetracyclic diphenhydramine, all molecules which I could have in that, and it predicted very well. And here you see the superimposition of these molecules of this pharmacophore with BS3, BS2, BS3. So there is 2 hydrophobic sites which you see here, and 1 is ionizable site. I'll show more clear will be. These are of course with this the predicted activity. Here you see the molecules number X7 is you are seeing 2 BS3 and BS2 as the hydrophobic site, and this is BS1 is the positive ionizable group, which was the nitrogen for this. And this compound very well fits onto these pharmacophores. And the distance you may see here, the BS3 and BS1 distance is 6.06 and it is 5.72 between BS2 and BS3. And here these distances. So this was the pharmacophore which was identified at that time, because we didn't have any structure-based design at that time, one time. Now a model is the one which predicts. Now prediction is very, very important. So it can say, so what I thought, what this we did a long back, in 1999, there's a paper on JMAT came for antihistamines. There was 4 molecules for which Ki values were there. I thought, how does my model predicts these activities? So when we predicted these molecules through our model, the activities are in IC50. So what we did, because that our molecule was based on IC50 values, so we tried to correlate, and you find it is a very good correlation. Even you take all the molecules, still it is R square was 0.6978. But if there was one compound not so fitting, it becomes 99, but doesn't matter, even 6978 for the whole compound. So this is the beauty of the model is. It means if you take this is I'm talking of 90s. And these are the models, these are the molecules of this Journal of Medicinal Chemistry paper of GMC 1999, which was predicted. Again, you see they were fitting very well into the pharmacophore. Now, thereafter in early 2000, the Apex model was discarded and the Catalyst came, and where you have these, I think it is Discovery Studio one, and you have these features which you have to match. I'll not go into detail for this. We put the hypogen because we had the exact activity data for this. In hip hop also the other one which one can use. The approach we have used extensively in our work later. And we took the best hypothesis out of it. And you see the compound, the pharmacophore which came from Catalyst was this. It was almost the same. You see here again 2 cyano, this is the hydrophobic sites. One is red one, which is a positive ionizable group. And this is the anionic side where the amide group comes. So this is the pharmacophore which came, and this means what we have analyzed A, B, C, D sites in our own 2D QSR, that hypothetical totally structural modify. This is what the sophisticated models for pharmacophore also gave the same thing almost. Of course, this is again another view of this hydrophobic site, hydrogen bonding acceptor, positive ionizable group. And this is the diphenhydramine molecule. And you know what that ortho substitution was doing in our molecules, but that it was making both rings non-coplanar. And this is what is there. Here also observed, you see the 2 phenyl rings hydrophobic that are non-coplanar. And this is what that observation was there in our 2D QSR, that there's a ortho position of the steric effect does something. And what does it? It does makes in ring non-coplanar. But then later on, H1 receptors, amino acid sequencing was known. And then I thought that we should do something. How does it is working? Still, I was having in my mind, there are 6 outliers. Why were they outliers? We could not get any answer from these models. It was saying the same. So at that time we did this, and we could do the modeling, homology modeling through bovine crystallized bovine rhodopsin, because that was the best. And you know, in the bovine rhodopsins, there are 3 loops with the extracellular and terminal. There are 3 intercellular loops, which are difficult to run, and 7 transmembrane helix. And then we developed this model, taking all these. I think this is all. I am also putting the references, which gives you the details of this. of the sequence alignment onto this. And you know, it also did not explain, it explained everywhere those things, but what happened, why still the outliers behavior were not explained, but in the meantime, in 2011, this human H1 receptor also came, exactly structure with doxepin. And when we compared our homology model receptor, H1 receptor with this, we found there's a distinct difference, particularly in the ECL2 loop, which is between TM3 and TM5, because it is elongated. In the one with homology, it was like a hairpin structure, while in the other one, it's elongated. And this was one of the reasons which later on proved to be very useful. Here again, you see the TM3 and TM5, this extracellular loop, and these are the amino acid aspartic acid 107X with positive ionizable group. This will also come in detail in the next slides. And this is how the doxepin, which was there, is minus. You see here aspartic acid 107 and 2 benzene rings are there, which are hydrophobic, where it's shown in Pharmacophore as well, and the ionizable, this oxygen, which is in diphenhydramine, and NSCO fragment, which goes with this threonine binds, and this is there. So what we did from this doxepin structure, we translated this structure-based information back into the Pharmacophore, just to see how our Pharmacophore, which we had developed, compares with the real Pharmacophore, from the real data co-crystallized with a human H1 receptor. And you see here, you compare these two, you have seen this one from Catalyst, Apex3 was almost also same. You see the distances. The distance in between the two cyno, which is hydrophobic HY2, HY1 in this, and you cyno here is 5.62, which is 5.6 there. The other distance is positive ionizable group, it is 6.32, here it is 6.4 on the other one, and you see this one which is a hydrogen bond acceptor, which was by oxygen, this is about 3.13, while it is 3.1, again there the distance is 3. So similarity. And this is what, if you rightly see the Pharmacophore model, it is not that you develop Pharmacophore computer, but which hypothesis you are selecting. That may not be the top one with a statistical point of view, but here you have to apply your medicinal chemistry knowledge into it. Then I looked into these 6 outliers, particularly these 2, 5 and 6. And this is how we say conformal mapping of compound 1, which was chlorothianol. We did it onto this again to cyno and this model. And we found that is a U-shaped conformation. You know, at that time it was U-shaped. We published that paper in JMedCom. It was a lot of questions came, but that also helped me in a lot in 90s when we were showing conformation, showing folded. But that was really folded conformation. And here you see the scoring of pharmacophores. Again, you see there are the 2 molecules which are there. Pharmacophore mapping of protein structure we are showing. And this is the yellow one and the green one. And green one, these are the sites, because here aspartic acid is 107, which is showing on this, and that is a positive ionizable with nitrogen, and there are 2 cyno rings. One this side, this is going on the other side. So these are the interactions with the receptor side include aspartic acid 107 and the tyrosine 108. This is for hydrophobic interactions, serine 111. So these are 428. These are 2 rings. So what happens actually? I'm not going into detail here. Here you see. This is the receptor picture, the docked compound, and this is the outlier one, 6, which was the most active. What happens is here is a static hindrance. If you put something here on the group, that cannot go. That's why it shifts into this. And the loop, which is ECL2, because of this elongation between TM3 and TM5, this creates a space here, and this binds, and this becomes a extra space for this. And that's the reason that you get an extra fine space where it binds. And you see these are the 2 which puts static hindrance, phenylalanine 424 and isoleucine 1,5 of TM6 and TM3, and this binds. So this is the extra binding site with these compounds who are showing far higher activity than what is predicted by our QSIR or even with Pharmacophore model, earlier ones. This does it. So this gave an answer to the outliers behaviors or those compounds which we identified as early as in 1980s. So in summary, the crystal structure of histamine H1 receptor differs significantly from homology model protein in the ECL2 region. And this beta hairpin is there, why it is elongated between TM3 TM5 to produce a ligand binding cavity. In the absence of receptor structure, we have postulated different binding modes for a series of these compounds based on ligand-based concept. Although our ligand-based concept was successful identifying a candidate molecule potent as, but certain perceptions regarding bulky substitutions at para positions of phenyl ring have remained mystery, which we could solve only with these later studies. Now, with similar studies later on, we have used, now we use extensively this focus library we make. I don't believe much into the databases, but still we did, because at one time in CDRI, after a long time, I was given an assignment for tuberculosis, and at almost in 2010 or so. So I thought, just to control by academically, administratively is not correct, I must synthesize myself something. So we identified this structure, which you see here, and then we made a focused library, and we thought that betaquilene was, at that time, was in the clinical trials. We designed it so Mycobacterium tuberculosis ATPase synthase inhibitors, and we designed these molecules, these libraries, and we got really very, very potent molecules. We filed the patent for them, and I think they showed inactivity in vivo. So I would say just making 15-20 compounds, we could reach into this because of this computer-aided design and virtual screening. Similar case came from, thus I'm saying that a drug designer, medicinal chemist can work in any area. He's an architect. The only thing he has to know is the knowledge of this. So again, we gave anti-Alzheimer's, anti-dementia, we make a model on the known molecules, then we identified the pharmacophore, then we make a focused library on the molecules which we can make, and finally we got also very potent molecules into which went into the patents. And that's why I hold so many patents, because very quickly a medicinal chemist who practices this in a real way can reach to the compounds of activity. Now I will just give you something about the repositioning, because COVID is going on, and for getting a quick answer, it is always repositioning of the drugs or repurposing. And you see here that these are the molecules which were for these activities, but new activities have been found from them to repositioning. And the same technique we are applying for COVID now, because we want very quick results. And I will not go into detail again. You know, the reason is that phase 3, you have to do only phase 3, phase 2. The rest of the thing, you know, the drug is already in the use and the human beings, so you can do repurpose. Again, I'll not go investigational new drug or emergency use authorizations, which are there, which are details. But the repurposing is very important. And because of getting this in the month of March, I got with something done on these. I thought what we can do. So we had these antiviral drugs, and we had also hydroxychloroquine, we were knowing, and we did some kind of a docking because we could get the structure of the protease from COVID, from this enzyme SARS-CoV-2, and we did some docking. And from this docking, when we sent this paper, it was not accepted because it's showing that we are not showing anything practical, but practical difficulty is there. How can you get them tested? In the meantime, when we were having this comments in Cell Research in 2020, in the month of March later, this paper came where there was some data available in vitro for this. So I immediately got it. One of my colleagues who works with me in the Global Institute of Pharmaceutical Education, this is Professor Sheshin Nandi. I said to him, you just see the correlation with this, and you see how our docking scores correlate better. And this totally describes this. And now I think it becomes a good model for us, and then we can just dock into the same manner and can find out where does the compound lies in comparison with remdesivir. Here you see the remdesivir was the best one on the top. Just to tell you, I think everybody should be knowing that we have these other molecules which are repurposed for COVID. Yesterday only I came to know that etolisino which we had at one time given a permission in India, drug controllers in 2011-12 for anti-psoriasis drug is now very much useful. Also, if it go with maintaining for, because it the storm of cytokines which takes place checks it, so it can be very useful in the last stages. And similarly, CDRI is trying umifenovir in phase 3 trials in our institute, and I hope that we should be able to come out with this thing. With these words, I thank you very much for your patient hearing. And there are a lot of people who have to be thanked because of the work, but I have always cited the references. So I thank you all for this. Thank you very much. Hello.
Hello.
Yes.
Yes, sir.
Yeah, now I've thanked. The talk is over. If there are any questions,
Yeah.
I shall be happy to answer the questions.
So, those currently.
Sir, am I audible?
Testing team.
Hello. Sir, am I audible?
Yes. Hello.
Sir, there is some technical problem. Please wait for a while, sir. Actually, some other people, they have gathered the question and they just, we have collected so many questions in the YouTube live channel.
Okay.
So just one more student, she is collecting and she gathered for the next question and answer session.
I thought, but I was audible now. It's okay.
Yes, sir. Your voice is very clear.
And actually, I took 45 minutes. I was told, so I finished in 5 minutes.
Hello, sir.
Yes.
Hello, Dr. Saxena.
Yes.
Very good account of a 3-decade or 4-decade-old story.
5 decades
And,
almost. Yeah.
I was watching you, but as I told that I promised to our team that I will not interfere much. Now Himakshi will take the question answer session. Thank you very much for accepting. It was very nice hearing you one more time. And Himakshi will now take over and she will be asking you questions. Thank you.
Thank you.
Hello, sir.
Yes.
Sir, we have received few questions. Sir, first question is, what is the optimum training and test set for QSAR?
Optimum test set. Optimum
Yes, sir.
training and test. Yeah, I would say that it is always good. The smaller is the training set and the bigger is the test set. But the optimum, I would say, one can go with start with 50-50% and can go to 33% training and 75 if it can go. But this is difficult. But if the model is good, very good model, if you compare two models in which one is having 50-50%, one is having 33% training and 75% testing, the other one will be called better. So it depends,
Thank you, sir. Next question is, what is your opinion on machine learning in drug discovery?
I think machine learning is a little faster as compared to the other one. And I think what I feel in both, whether it's a machine learning or the other algorithms, both work. Only thing is the person has to, what I would like to emphasize, which I have emphasized, the persons who use these should know also a good medicinal chemistry. If you have a good knowledge of medicinal chemistry, model you derive, which model you choose. This is also very, very difficult. Difficult in the sense that you have to have background. I have attended so many conferences outside. People are extraordinarily good on computers. But what they are giving out, is very difficult. They are considering one terpene structure is coming because they are not going what is this structure, what is dopamine, where is this thing. No, just because it is shown, they will show. So I would say that the judicious medicinal chemistry background is most essential, but I think machine learning is faster and one should take advantage of machine learning, which is coming up.
Thank you for the answer, sir. Third question is, what is indicator variable?
Indicator variable, yeah, very good question. I use indicator variable very extensively. If there is something by which you cannot parameterize, it's like a joker in the play cards. So you can give it as a substitution. Suppose in a structure, there are diverse structures. So you can put indicator variables for absent and presence. And if that comes, that is giving a kind of a mathematical orientation to your observation. Okay. And the other thing which indicator variable can do sometimes in enantiomers, when there are two enantiomers, DL and racemic, you can put the value for one enantiomer. You cannot have other property, something which you can distinguish. So you can just give a indicator variable I+1 for levorotatory, -1 for dextrorotatory, and plus just 1 for racemic. Yeah. And you can get the answers, predictions on in the equations you can generate. So indicator variable is the variable which is on your choice, the way you want. This is a very, very good thing.
Thank you, sir. So fourth question and the last question we have, how can I select best descriptor in QSAR model building?
Yeah, that's very, very, very important question, because in today's scenario, you have sometimes 30 molecules or data set of 80, and you have got 2000 variables. Which variables to select? That is a multi-million-dollar question. But I think here again, I would say the first thing one should do, and people are doing in the analysis also, which do the kind of a principal component analysis, they find that many variables will be giving the same kind of information. So one has to firstly cluster them in any method they can use statistical methods, or the principal component can use component, or it's also partial least square analysis. That kind of a thing can be done. So you can, number one, you can reduction approach, you can reduce them. But again, there you have to think in terms of medicinal chemistry again, what is the utility of, what are the parameters, what those parameters giving and what you want. So that is again a very, very crucial step. And crucial step, and here you need very judicious thinking. It will not go just blindly that, okay, you select this here, you select that. If suppose you have got a positive ionizable group, you know aspartic acid is one of the very important amino acids which bind most of these GPCRs. And suppose you are having some parameter, you have to give that there must be some parameter which should give indication for that kind of interactions. There must be some parameter which should give hydrophobic kind of interest. So I think that is the choice you have to think in that manner.
Thank you, sir. Sir, on behalf of DDH training team, we would like to thank you once again, sir, for being with us. We hope that your talk will motivate the participants to participate in Drug Discovery Hackathon. Thank you, sir. Now, sir, we will leave this session here, and I request all the participants to join us again in MS Team or YouTube by 4 p.m. for Vertical 2 session.